Hello everyone, welcome to Healthcare Without Harm Europe's webinar series on pharmaceuticals in the environment. My name is Adela Magyar, I am the Pharmaceuticals Policy Officer at Healthcare Without Harm Europe, and I will be your moderator today when we talk about the need for a new strategic approach to address pharmaceuticals in the environment. We have over 90 people from over 20 countries that registered to participate in Healthcare Without Harm's webinar on pharmaceuticals in the environment today. We are delighted to have all of you, and I'm sure this will be worth your time today. For those of you who don't know Healthcare Without Harm, we are a non-profit membership organization of hospitals, healthcare systems, healthcare professionals, local authorities, research institutions, but also environmental and health organizations. We work to bring the voice of healthcare professionals to the European policy debate about key issues such as chemicals, climate change and health, green building, sustainable procurement, pharmaceuticals, antimicrobial resistance, sustainable food, and also waste management. We also work to educate the healthcare sector to understand the importance of the environment and become leaders to advocate for broader societal policy and changes. Our mission is to transform healthcare worldwide so that it reduces its environmental footprint, becomes a community anchor for sustainability, and a leader in the global movement for environmental health and justice. Our main policy areas are listed in this slide. We currently have 88 members in 25 countries of the WHO European region, including 16 member states of the European Union that are committed to the mission and goals of Healthcare Without Harm. At the same time, Healthcare Without Harm leads the Global Green and Healthy Hospitals platform. This is an international network of hospitals, healthcare facilities, health systems, and health organizations. Members are dedicated to reducing their environmental footprint and to promote public and environmental health. Also, Healthcare Without Harm Europe leads the only EU-wide campaign on pharmaceutical pollution called Safer Pharma. The main aim of this campaign is to make the EU the first region to legislate on pharmaceuticals in the environment, so please join us at saferpharma.org. In terms of our webinar today, just a few premises. The presence of pharmaceuticals in the environment is well documented. Our speakers today, in their early publications, state that pharmaceuticals are already damaging the environment and, in the long term, they could cause widespread damage to human health. However, at the EU level, pharmaceuticals in the environment are mainly addressed in legislation of medicinal products, but not fully tackled in legislation on medicinal products for human use. The European Commission's long-awaited strategic approach on pharmaceuticals in the environment is meant to cover this legislative gap, but it's already more than 3 years late, and it is increasingly unclear if this long-overdue approach will be published under the current Commission, despite a call for its swift publication in a letter consigned by environmental officials from 10 different countries. Our webinar today includes 2 speakers. Our first speaker is Professor Klaus Kümmerer, who is a professor of sustainable chemistry and physical resources at the Institute of Sustainable and Environmental Chemistry, ISEC, at the public Leuphana University in Germany. As the chair of ISEC, Professor Kümmerer's research and teaching is focused on sustainable chemistry, sustainable pharmacy, material resources, aquatic environmental chemistry, and environmental and sustainability research. He received the Recipharm International Environment Award for his work on pharmaceuticals in the environment and the Water Resource Award for the Rudiger Kurt Bode Foundation for his outstanding work on the benign by design concept. He has published extensively on these topics in leading peer-reviewed scientific journals of different scientific disciplines, has many book contributions and also co-edited many books. He is a founding editor of the scientific journals Sustainable Chemistry and Pharmacy and Current Opinion in Sustainable Chemistry, as well as associate editor of Chemosphere and Environmental Pollution. Klaus Kümmerer serves in many international committees, including the prestigious seminal Global Chemical Outlook by UNEP, the EU Technology Platform, SUSCHEM Europe, the Commission for Water Research of the Deutsche Forschungsgemeinschaft, the Executive Board of the Subdivision of Sustainable Chemistry in the German Chemical Society. He is also the scientific organizer of the annual Green and Sustainable Chemistry Conference and the International and Interdisciplinary Summer School on Sustainable Chemistry. Our second speaker today is Dr. Tim Ausderbeck, the head of Water Resources Management at IWW Water Center in Germany. Tim Ausderbeck studied hydrology in Fribourg and also in Vancouver in Canada, and was previously employed at the Center for Environmental Systems Research in Kassel and Heidelberg University, where he received his PhD degree. Since 2012, Dr. Ausderbeck has been in the IWW Water Center in Mullenheim, where his current research questions deal with organic pollutants in the aquatic environment, climate change impacts, water resources management, and water quality issues in general. He has acquired multiple research grants from visitors, from various national and international organizations, including EU and UNEP, and is also an author of 25 water-related publications. There will be time following the two presentations for questions and answers. All participants will be automatically muted during this session. The webinar is being recorded and will be made available on Healthcare Without Harm Europe's website over the coming weeks. At any time during the webinar, if you encounter problems with your audio system or you have a question for a speaker, please write it in the chat box at Questions for Healthcare Without Harm Europe. So again, welcome to all of you and to our distinguished speakers. And with that, I would like to give the floor to our first speaker, Professor Klaus Kümmerer.
Yeah, hello everybody. This is Klaus Kümmerer, and I'm glad that I have the opportunity to contribute and to disseminate some of our findings and our views, and hopefully they are helpful. It's very short, about 20 minutes, so I will not go into details. So, well, I'm presenting first a little bit of a background. I guess most of you you have already the knowledge, but just to set the theme. And also, I think it's important that we think about solutions and not only about problems, and that will be the second part of my presentation. For more information, you see here on the first slide the links where you can get more information. After short introduction, I will present you an approach of what we are doing and what is called benign by design, with a focus design of pharmaceuticals and chemicals, by the way, also used in medical environments and other chemicals. What is the approach of that? A few examples and then the conclusion, take-home message. Yeah, I think the speaker after me, Tim Ausderbeck, has done a nice study with some colleagues, and what you can see here, pharmaceuticals are a global issue. They are more or less everywhere, and where you have gray colors, that does not mean that pharmaceuticals are not there, but just not measured. And I think the same holds also for the colors. The more intensive the red just means more research and more analysis has been done. So it's a global issue, pharmaceuticals in the environment, pharmaceuticals in water and soil. Here an example from Germany. So we find, for example, metformin, an anti-diabetic, in rivers, in River Elbe, close to Hamburg, as you may see here, and in another river, and even in the North Sea. And what you also can see, the black bars That's a transformation product, a product of incomplete biodegradation, and that's important. We not just have to think about or should talk about degradation. We have to specify what do you mean, and I will come back to this later. What this slide also shows, that and the previous one, that even if there's a effluent treatment in place, conventional effluent treatment, compounds are not removed at all, or at least not removed completely. And some compounds are transformed in the environment or might be transformed in effluent treatment, conventional effluent treatment. So there are many examples of that. And then the idea was, as another end of the pipe approach, okay, we need additional so-called advanced wastewater treatment. And also here you see an example, ozonation, one of the examples using chemicals for oxidation of parent compounds. And what you can see, there are compounds like paracetamol that are nicely removed. I do not say mineralized, it's just removal. That means they are not present in the water phase or detectable in the water anymore, but maybe they stick to the sludge. Or, and I will give you some examples later on, they are transformed to follow up products we have not yet measured or we will not be able to measure. And another, in the bottom part, you see a combination of treatment that is using powdered activated charcoal in combination with ultrafiltration. And you see, okay, paracetamol is nicely removed, but sulfamethoxazole, for example, is not removed as well. And what you also can see, there is a huge range. So obviously the rate of elimination depends on the specific conditions of the treatment or the type of treatment you're applying. And on the right side, in both cases, you see there are also obviously compounds that are not removed at all by such a treatment. And please be aware, you also need energy for such additional treatments for pumps, but also for the chemicals. Ozone is a highly reactive and dangerous chemical, has to be handled accordingly, needs energy to synthesize this. Also, charcoal or activated coal has to be synthesized and has to be removed again. And the membranes for ultrafiltration have to be manufactured. And again, you need pumps to press the water through it. So just think about how could this work on a daily basis for 100,000 cubic meters, for example, which would be a size of a sewage treatment plant in Germany of about a city of 600,000 inhabitants. Also, we have seen there are many precursors, often named parent compounds. Maybe you have heard of methadone for treatment of addiction to drugs, to illicit drugs. It's a precursor as other drugs containing amino groups. With chlorination, which is in some countries applied, will lead to a compound called NDMA, nitrosodimethylamine. This compound is highly hepatotoxic, and it's probably a human carcinogen. So there's a threshold value set in the USA by the US Environmental Protection Agency, 7 nanogram per liter. You know the unit gram, so 1,000th of it is a milligram, another 1,000 is microgram, and another 1,000 is nanogram. So 1 billion nanogram per liter. It's a limit that should not be exceeded to be on the safe side. Another example is on the following slides from our lab. Ciprofloxacin, we studied here, a very well-known antibiotic. And this antibiotic is very stable in the environment. So, and we have often stabilization, hygienic stabilization, originally of raw water, potable water, by treatment with UV light to kill the microorganisms. But it has also been under discussion for removal of micropollutants, including pharmaceuticals. Here you see an example, the black bars. This you see ciprofloxacin after 2 hours of treatment in the lab is nicely removed, and also the antibiotic activity is more or less gone. Then you have a line on the upper part. On the one hand, that is DOC, dissolved organic carbon. That means they are not the compound specified, but these are organic chemicals. And these, what you can see here, is not changed anymore. That means the parent compound was eliminated, not detectable anymore, but obviously the organic carbon linked to it is still present. And what you can see below all the bars along the time axis is a huge dynamics of different compounds. Each bar is another compound. High bar does not mean that it's in high concentration, or low bar does not mean in low. It could also be the opposite, because these are totally new chemicals that we cannot buy, and therefore we cannot quantify it by our analytical means here, high resolution, LCM, SMS. So it just tells us there's a dynamic in there during, and it depends on the treatment time, what you get within your treatment. And the graph also, coming back to the DOC, tells you, in fact, maybe ciprofloxacin is removed, but many other compounds are formed, and DOC is not reduced at all. And if we have a look on toxicity, what's going on there, you can see there's a window of genotoxicity, in this case measured or monitored by the micronucleus test. These are the bars, and you see within a certain time window, there's also a window of genotoxicity. So again, it depends on the time of treatment, whether you have a detoxification or whether you might have an increase in toxicity of the treated solution. So in fact, the advanced effluent treatment has several limitations, and we come up with many, many new compounds. And if you think about for mutagenicity assessment, for example, we have 4 to 5 different Ames tests in 2 versions, so that's already about 10 tests if you want to go for complete assessment. We have not yet tested on genotoxicity, neurotoxicity, endocrine disruption, etc., etc. So if you have one compound and you come up in advanced treatment, and to a lesser degree that can also occur in conventional treatment, so incomplete biodegradation resulting to transformation products. Then we have 5 compounds, and as I said already, let's say 10 endpoints, not yet taking into account the aquatic toxicity endpoints. You may easily end up soon with 50 or 100 tests. That's too expensive, and that takes too much time. That will never be feasible. And also, we have about 3,000, a little bit less, but around 3,000 active pharmaceutical ingredients on the market. So you can easily calculate. And for testing, for transformation product testing, you need the compound. So all of these you want to test that you might have been able to analyze, and we do not even know whether we got all within analysis. All that you have analyzed and you are able to establish at least the provisional molecular formula, you cannot buy, so you need to synthesize them, and roughly 5,000 to 10,000 euros per compound. Someone recently has calculated that would be several billion euros only for synthesizing a certain number, not all, but maybe the majority of transformation products we know nowadays. So another point is, as I told you already, we have also metabolites. From medical literature, we know a little bit about them, but very often, if they are not the main product of excretion, then it's just mentioned without further information, maybe sometimes about cancerogenicity because of bladder cancer, but that's very often more or less, and nothing else is known. And these technical transformation products I talked already about, and also if parents compounds, and we know also examples if transformation products from treatment go into the environment, there are also natural processes like hydrolysis, oxidation, photolysis, reduction that can lead to new unknown chemicals or products or pharmaceuticals. Some of them might be still active, some not. Some we know have been shown they are less toxic, but there are also increasingly numbers of publications demonstrating that these products of incomplete mineralization might also be even more toxic than the parent compounds. And we do not know what that means for the future. Compounds are undergoing a dynamic, and you see here some data from a German Environmental Protection Agency. In blue, the compounds, some of them, their share in 1999, and the change until more or less within 1 decade, the data from 2012, and you see there is a huge change, and interestingly, very often we, the scientists, study the compounds of the past, and we do, as we have seen, each chemical behaves, or pharmaceutical behaves different, so probably we will not be able to make any far-reaching predictions about the future and the treatment. What we can say probably also with the new compounds, there will be transformation products of incomplete degradation, and probably some of them will also be more toxic or will be circled in the aquatic cycle. So summarizing half-time, I would say there are clear deficiencies of effluent treatment, advanced effluent treatment. So technologies are emerging, but even combinations fail to remove all contaminants. The efficiency strongly depends on the type of compound, and that's also quite clear because each molecule is a different character, I would say. Therefore, they have different activity, different water solubility. So if we talk about pharmaceuticals in the environment, we talk about 3,000 compounds, but they are individuals, and grouping is very often not the best way to handle them. Very often we need to go on a case-by-case basis. So I also mentioned that advanced oxidation processes, AOP, example was ozonation, result in reaction byproducts that may be toxic, mutagenic, and so on. We have to care for increased costs. So, and there are data out there, but it's not clear what is included. Very often it's just the cost for the chemicals or for the energy, but you have to enlarge your plant, you have to synthesize the chemicals, you have to go for resources, all that. And you also have to know, in many countries, if there's a heavy storm and rain event, then most of the water, the effluent, will circumvent the sewage treatment plant, because else all the bacteria of the microbiological stage would be swept out and the plant wouldn't work anymore. So there is no treatment, and there are data saying about 30%, for example, in Germany, of wastewater throughout the year is not treated at all for that reason. And there are data from the US and from Germany indicating that we have an increasing share of piping in the underground that is broken, between 20 and 50%. So there is also water, effluent, wastewater going directly into groundwater without any treatment. And we should not be too, how should I say, Western focused. According to a report of United Nations Water, 80% of the world's wastewater released is released untreated. Not even one stage or conventional treatment, let alone advanced treatment, let alone the cost for poorer countries and the knowledge to have such advanced treatment. At the same time, most often these are the arid countries increasing, or maybe probably more exposed to the climate change. So, and there we also have population growth more than in Western countries. So there is an increasing need of water, and therefore also an increasing need of reuse of water. That means also we need cleaner effluent. And cleaner effluent we can get only if we have cleaner influent. So we have to go from the end of the pipe to the beginning of the pipe. So that's the second part. What is the approach? We are all very good in solving problems, but I think if we can avoid problems, then we will avoid all the issues I have mentioned before: costs, transformation products, etc., etc. Sounds a little bit strange, or and probably some of you are very skeptical, and it took us a long time to come up with good examples, but it's feasible. And that I will demonstrate you what is the problem. The problem is persistence. And if we avoid persistence, then by fast and complete mineralization in the environment, then we will have no exposition, no effects, and no risks. And that sounds a little bit strange, but the point is that very often medicinal chemists, or at least the CEOs of the companies and the sales managers, they use the term stability. A compound has to be stable to be successful on the market. But that's not a scientific expression of view on the topic. A scientific, pharmaceutical, or chemical view would say, what is stability? Stability means how long does it take until a compound reacts? How long is the half-time? And if you have that view, then that brings into your mind under which conditions. So if it's, and then you see, for example, the excess of light is different. And if you see also, if it's on the shelf or if it's in the environment, also the bacteria in the gut are different. The pH in your stomach is different from what you have in sewage treatment or in surface water. So we should talk about reactivity and adapt this times, half-lives accordingly. And that there are already so-called unstable pharmaceuticals out there. For example, some years ago, my children really needed an antibiotic. We are very careful, but they needed one, amoxicillin, and we got a powder from the pharmacy. And then there was stated, yes, mix it with water and then put it into the fridge. Why putting into the fridge? Putting into the fridge because the temperature is lower and therefore the half-life for its degradation, for its hydrolysis is much slower. So unstable pharmaceutical, very successful on the market. And maybe you already have got some pharmaceuticals in brown glass on a brown blister. Why that? Because there is no light access. So these compounds are sensitive against light. But in the environment, we could use this if we have such compounds, and if we make sure it's not just transformation by light, but it's complete degradation. That means mineralization. So some examples to show you that's feasible. Here an example from our research, especially the reference is the lower one, ES&T, was editor's choice, and therefore you can load down its open access. So a beta blocker, propranolol, we treated it by light and were a long time interested in the stable photoproducts that were resistant against biodegradability. And someday we came up with the idea, if there are biodegradable transformation products generated by photolysis and they are still active, we might come up with a biodegradable drug. And that's what we did, and you see here the scheme, how we did this. So we came up in the end with a very similar compound for hydroxypropranolol. And that's has more or less the same. We did then an assessment, synthesis assessment of its biodegradability, but also even more important, we were able to establish or to show that it's still active. You can see here in an in vitro, in a lab-based test with living cells. So the EC50 or the effectivity threshold 50% is 0.31 micromoles. That's a little bit lower than the parent compound, but it's more or less the same range. It does not really matter. And also, as for unwanted side effects, they have to be absent, else you will never have a drug. And and we could demonstrate, and I will not show this here because of time, that this new drug, this new active compound, is in no case less good or worse than propranolol or two other closely related compounds, metoprolol and atenolol. So we have a new active compound here, and that's also a new opportunity, market opportunity, business opportunity for companies. Should be. And that's not our only example. We have, I'm not yet finished. Two final slide and conclusions. Lloyd? Oops. Sorry, it was my mistake. Still present. Sorry. So redesign and de novo design. Redesign was the beta blogger example. We were able, together with the German Center for Anti-Cancer Research, to come up with a new active principle in active cancer treatment, now in clinical trial phase 2. Only recently we filed 2 patents for antibiotics, derivatives of ciprofloxacin, which is a very stable and persistent compound. And on the bottom right side, you see an example of chemicals, ionic liquids, also demonstrating that it's a broadly applicable compound. That's good, but first of all, and that would be a different approach and a different presentation, make sure if you're going to the beginning of the pipe, not just the molecules, if there is no need to use a certain compound, a certain pharmaceutical, that's even better. So first think about if it's needed. If yes, then we can go for a redesign and completely degradable compounds. And there are, for example, OECD data showing that there's a huge span in Europe, for example, using antibiotics. In the Netherlands, it's only 1/3 than is used in the candidate, with in Europe using most of them. So I guess the people are more or less the same in different European countries, but we could reduce antibiotic input into the water by about 2/3. And that should be the very first point, and that's also part of a green and sustainable pharmacy, also thinking first of that and then thinking of benign by design for new compounds. So the take-home message is, I think I could show you environmentally benign design is feasible, and that's a good approach to reduce micropollutants, pharmaceuticals share of them, but also others. Inherently, that's very important. There's no technology needed. We applied biodegradability tests that made sure that it's not only degraded in a sewage treatment plant, but also in surface water. And I think that's urgently needed for other countries where there is no effluent treatment in place. And for the companies, I think this approach comes up with new compounds, and it's also a new business opportunity. Thank you for your attention.
Thank you, Professor Kümmerer, for your presentation. We will take questions at the end. I give the word now to Dr. Tim Austerbeek.
Hello, everyone. This is Tim Austerbeek. I will show you a little bit, or build a little bit on that what Klaus Kümmerer said. So I want to show you that's next to the benign by design concept, there's also another opportunity for you to avoid pharmaceuticals entering the environment. And I just want to show you one pilot we did in the region here. And so we call it keeping the river clean, because we just want, similar to what Klaus said, we just want to avoid the chemicals, the pharmaceuticals, to enter the environment. And that is, of course, a big task, because as we've seen, it's very expensive in the end to do the end-of-pipe treatment. So we should focus on, not even letting those getting into the environment, so that we can also save money by that, and that we spend that money otherwise in a better way. So Klaus already showed that slide. It's a publication of mine 2 years ago, and what I just want to show you as a motivation is that we have a couple of hundred different pharmaceuticals in the environment, and in those gray countries there are no measurements, but I'm 100% sure that you will also find pharmaceuticals in the environment. So the question is, how can we on a larger scale prevent this? How can we mitigate this? And coming back to the sources of how do pharmaceuticals enter the environment. So we have the consumers, we have the industry, we have agriculture, aquaculture, and the hospitals. And in this pilot study, which I'm presenting here, we want to focus on the consumers and on the hospitals. So, and we also want to concentrate on one specific group of pharmaceuticals. These are the iodinated contrast media. I'm not going to read all this, what you can see here, but I guess most of you have heard what it is, or even taken it. If you, for example, had to take an X-ray at a hospital, for example, or at a clinic, then you probably had to drink a little cup of this contrast agent. And when they take the X-ray afterwards, they can actually see how the contrast agent is moving through the organs of the body, for example. So, and what are these ICMs, this contrast media? What do we know about them? So we know that they're very difficult to remove at wastewater treatment plants without advanced treatment. And if you remember that one of the first graphs that Klaus was showing, where he showed all the different kinds of pharmaceuticals and how bad or how well they are eliminated in the different advanced treatments, you know, with activated carbon and ozonation, there were most of these ICMs, they were on the right-hand side of it, which meant that the elimination was quite bad. And so that we really need another option for this end-of-pipe method treatment, so that we need to avoid it getting into it. And the other problem is that in many regions of the world, also here where I live, in the Ruhrgebiet area of Germany, there is a problem that you take up the surface water as drinking water. And as these contrast media were designed that they do not absorb to the body, then that means you have a high excretion rate. And that means that these contrast media go through the waste. So after excretion from your body, after you've taken them, they go to the wastewater treatment plant. They cannot be very well eliminated there, and then they end up in the surface water. And there, from the surface water, you take it up again as drinking water. And we have concentration values. We have thresholds. Different countries. In Germany, for example, drinking water is 1 microgram per liter. And I'm showing you here in the picture the Baldner Lake close by here in Essen, and even one application in the hospital, for example, in the area of this lake, it will lead to a concentration of about 10 nanogram per liters. And that's just one application. So that means we have some problems here and we need to avoid that. And then coming to the next slide, it shows you the concentrations from the different contrast media we've looked into. And you can see this chart as going from the left to the right. On the left would be the start of the River Ruhr, starting in a very rural area, not many people there, and then going through the very industrialized area of the Ruhrgebiet area, from Dortmund to Bochum, Essen, Mülheim, and then finally to Duisburg, where it goes into the River Rhine, largest river of Germany. You probably know it. And you can see that the concentrations are increasing and even above thresholds, which we know, so above at least some thresholds. And the question is, how can we avoid that, especially then if we want to drink the water afterwards? And coming to the next slide is, I want to show you how exactly the contrast media gets into the Ruhr River and thus in the drinking water. We have the hospitals where we have the application, and from there the wastewater, if a patient uses the toilet there after ingesting the contrast media, then it will go to a hospital wastewater treatment plant and then to the Ruhr River. Or sometimes the hospitals don't have their own wastewater treatment plant, and then it will go to a municipal wastewater treatment plant. But also many times if you take a dosage of the contrast media, that also means that for the next few hours, whenever you will use the toilet, that there will be an excretion of contrast media. So that means even at home, when you're at home, you will have this. So this makes it very difficult to control this. Especially at home, if you use it at home, then it goes quite sure to the municipal wastewater treatment plants. So in the end, we have two different targets. So we want to protect the environment, meaning we want less of the chemical in the environment, in the Ruhr River. And we want to protect the humans, because we want to have less of the chemical in the drinking water. because it's very difficult, as I said, for the wastewater treatment plant. It's already difficult there to get it out of the water, to eliminate it, and this also holds true for the drinking water. So the next slide shows you again the circle on the left side, which I meant. So you use it as a patient, then you have the excretion, and there we want to, get into it and want to do a urine collection. So that means that the urine, which has high concentrations of this contrast media, doesn't go to the wastewater treatment plant and then serve as water and drinking water. So we want to break the cycle there. And that's why we focus on the hospitals and on the doctors. So in the end, what we want to do is that we want to collect the urine, and we want to, therefore we need to design a communication strategy and, newspaper, television, and so on, because it's very important to inform the people. Then we want to test it, or we did what we did in the last year, we tested it, and then we did a practical application in our city here, the city of Mülheim an der Ruhr, with about 180,000 citizens. 2 big hospitals, 2 practices where you are given the contrast media. And there we asked them if they could be our partner and they could help us, to do the urine collection. And in the end, we also wanted to see how much money would it cost if we would do that on a larger scale. So we have 6 key messages from the study, and the first one is that the collection concept was successfully implemented. So we had 87 or up to 87% of the participating patients were using the urine bags. So that is quite good. We didn't expect that because it can be a delicate issue for many people. And then we try to come up with some catchy and funny slogans, and not all of them translate very well into English, but I tried a little bit here. My pee goes into the garbage bin, or the bag for the small business. And we handed those out, those postcards and those kits where you had 4 urine bags in there, and we had these explanations in 5 different languages. And then we were looking for the feedback, so with these funny postcards, but also online forms, and we also called about 60 people of those, and I was quite happy that it worked out that well. So the second key message is that you need medical partners. That's very important, because that's where the direct interfaces with the patients. So, and the patients also strongly believe in what the doctors say, and if they support this work, it's also helping the environment. And this is something which we also learned during the project, that it's very difficult, because scientists and doctors sometimes speak different languages. Many doctors didn't even know that there were environmental implications of medicine. And this is something I think which should also be done on a larger scale, also to inform doctors. So what was the effect of the collection phase before I come back to the other key messages? So what we saw there was that we saw a decrease of concentrations. We're quite happy about that. Here you can see 5 different contrast agents, and there you can see that for some of them, or for most of them, we saw a decrease during the urine collection campaign in the wastewater treatment plant effluents. We didn't expect this because the wastewater treatment plant is quite large, and Mülheim, the catchment of the clinics, makes up only, or of Mülheim makes up only 30% of the wastewater coming into that plant. So, but even then, we still saw some effects, which was quite well for us. Also, the load of the contrast agents decreased, as you can see on the right-hand side, where you see the influence and the effluence of the wastewater treatment plants during urine collections and without urine collection, like a controlled phase. Then the third key message is that our project Merkmal was quite successful, because the concentrations went down, as I just showed you in the map. On those graphs. I'm not going into details now, the consumption and concentrations, but you can contact me if you have, or ask any questions afterwards. Then we thought, okay, how much kilos, or how much can we save of chemicals entering the environment, and we did a rough estimation of the 4 months of urine collection, and we saw in those 4 months, we saw about 300 kilo, which we would calculate to the entire year of total application. And in the worst case scenario, only 19% were actually using the urine bags. So that would prevent about 60 kilos. In the best case, it was 270 kilos. And in the weighted case, where we looked at the patient demand and participation rate per institution, we came up to about 210 kilos. That's about 2/3 of the total input into the environment here. That's similar number to what Klaus Kümmerer earlier said with his method also. So this is a similar number. And the next key message is concerning the costs. So the additional costs for using these urine bags after the examination, this amounts to less than 10% of the entire treatment. So this is much more feasible for us than spending more money on additional water treatment in the end. I'm sorry that this graph here is not very well, this table is not very well presented. But what you can see is that in the end, a kit of 4 urine bags costs about €3.20. Then you have some other costs like personnel disposal and so on. And that makes up about €5.40, €6.10, depending on the practices on the hospitals. And if you compare that to what it costs, to have a health insurance based medical examination, that's about €50 in Germany. So that makes about 11%. And of course, if you have a private medical examination, it's more expensive, then it's even less. So for us, in our opinion, this is a very good way ahead to deal with that. Then the other key messages are that it was very important also to provide information material to patients and also to medical practitioners, because the patients were very interested in the project and they developed an environmental awareness. And now, even 1 year after the project, we still get calls and get emails and asked how the project is going and if they could do any more work on that to help us and if they can support us in any way. In the end, we say that the final key message is that it is a very proposing method for the future, not only for this pilot area, but also for larger areas to move on with that, so that we can, avoid these chemicals coming into the environment. And our plan is to build next year to have a big project on that, not only looking at the Mülheim city, but looking at the entire catchment of the Ruhr River, which is one of the most densely populated areas in Germany. So that's quite a big approach. And we have 9 big cities with about 29 hospitals and 58 practices where contest media are provided. So this would be a huge rollout for us. And I think what I forgot to mention in between was that if you ask yourself what's happening with these urine bags, so it works that if you use them, then the urine is used by a super absorber, and it binds it drip-free and odorless, so that in the end you have a hard gel which you can just put into your normal garbage bin, where it's, at least in Germany, it's usually then burned afterwards. So, and here is a graph just to show you a picture of the new project we are planning on a larger scale. So that would be the blue one, that would be the Ruhr area. And in the end, our idea would be, if that works well also, it would be our idea to do even larger rollouts also to other countries and also other states in Germany. So whoever is interested, we would be happy to work together with you also to provide our materials and to help this grow and to reduce the input into the environment. Many people were involved. If you just look at the, at the sampling, all the different ministries and everyone involved. So I want to thank all of them to making this study possible. And we also have our homepage. You can read it here, merkmal-ruhr.de. And it's unfortunately it's only in German, but please let me know if you have any specific questions. And I also hope that we can put this this video now, which we are taping right now, that we can also put this on the homepage. Of course, if healthcover.harm is fine with it, so that you can also see it at the homepage. And that's it. Thank you very much for listening.
Thank you to both speakers. It's now time for a Q&A sessions. We have received plenty of questions from all the participants, and we will answer as many as you can, but we will make sure that the speakers receive your questions and will follow up with you. So the first question for Professor Kümmerer is the following. Ciprofloxacin as well as other antibiotics may drive antimicrobial resistance while they are in a wastewater treatment plant. Have you tested or know about this type of scenario? Concretely, during the treatment, can be interesting modeling this type of potential resistance while antibiotics are inactivated.
Thanks for the question. We had already 20 years ago some studies using conventional microbiological methods, and there was already indication, and that was nothing surprising, that resistant bacteria against antibiotic resistance are in sewage treatment plants and in effluents. And it was also that the difference between sewage treatment plants where you have no hospital effluent on the countryside and municipal ones was not really striking. It was only a little bit, if at all. And the same, by the way, holds also for the compounds. So often we think the most important source are hospitals for antibiotics and other pharmaceuticals, and lots of work from several groups in several countries show that's normally not the case. It's only a minor share. There might be one or two compounds that are not, which you cannot take orally. There's a difference. And in the meantime, there is a lot of literature out there demonstrating that antibiotic resistance material and bacteria are present in sewage treatment plants, are present in influent and effluent, and there are indications that some of the advanced treatments even select, that does not mean enrich necessarily, but select some of the resistant bacteria, maybe because these type of bacteria are less accessible to advanced treatment like ozonation. So that was for us, again, another point to think about how can we tackle the issue at the very beginning. At least that there are no active compounds present anymore that contribute to selection of resistant bacteria in the environment in sewage treatment plants. But most of the resistance that we find is generated just by use and application of antibiotics. So we have to think about a proper use, and I just decided that I will modify my presentation a little bit. I will insert a slide or two which demonstrates, which shows clearly that we use more antibiotics that are needed for reasons of health. And probably some of you are living in countries where you can buy antibiotics over the counter. And that's one of the worst things you can do. Antibiotics should only be available by prescription. That means only in case where it's necessary. Even in our country, we have cases or patients go to the doctor, they get a recipe or a prescription, and they are glad, and it was a good visit to the doctor, if they come home with a tiny bag and there is something in. And then sometimes they do not use it, or if they use, then they do use it sometimes in the wrong. That's one point. And then they store it, and later on they flush it down the loo. Yeah. So that's one part of the story. The other part is a lot of people in the northern hemisphere. We have now autumn. They catch a cold and they think, oh, I need an antibiotic. But most of the colds are caused by viruses, and antibiotics are not active against viruses. So we have to inform the general population more about this, and often also because of lack of time, people, when they see the doctor and it's clear there is an infection and you need an antibiotic, they prescribe a broad spectrum instead of having a tough and good microbiological analysis that will take a few hours, but within these hours you will not die, and then you can select the right and the most effective, and that also antibiotic, and that also contributes to a reduction of the resistance. So again, summarizing, resistance is present everywhere, and the most important reason is the usage or the not correct usage of antibiotics. And if we have compounds that are nicely and quickly completely mineralized, then we tackle this problem also.
So Professor Kümmerer, another few questions for you. Did one of the substances synthesized by you received already a market authorization? If not, why not?
None of these compounds did. For the beta blockers, for example, we did a very thorough search beforehand, and then we came up with this compound, and then we found out that it has been described in literature 40 years ago as a metabolite which could be active. And then it was not possible to patent it. And if it's not patented, then probably no company is interested maybe to have it as a example. Maybe a foundation or even a state will go for it just to have this example or to release their waters. As for the antibiotics, we have the patent, but and we are now at the stage, we have an active compound, and now we have to follow up within the normal process that has nothing to do with biodegradability in the environment, with the normal to develop it to a drug, and that takes some years at least. So unfortunately, we still have to be patient, but the important point is we could show mineralization, degradation in the environment does not rule out drugs and active compounds that could be used in the future. That's the important message here.
Another question is the following. What is the timing to achieve the ideal situation to reduce these 2/3 of pharmaceuticals by reducing the persistence of the drugs? And what would you suggest to do in the meanwhile that we get this reduction? Would you recommend the effluent treatment as a transitional solution?
I would not recommend the effluent treatment, the advanced effluent treatment as a transitional solution, because that means you have to have big investments. And that means the technology will be in place for a longer time. It's not just in, out, on, off. And but what we can do, and that was a little bit unclear as the question, what we did with the biodegradable drugs, that is not that we can reduce it by the ones that are used by 2/3. That is before that already, by proper prescription, proper information, different cultures, and so on. As I already said, I will include the slides in an update of the slides. That we can start now. We can start already. Think about when is it necessary, and that there are data also out, and I will include this also, not just for antibiotics, also anti-diabetics, psychopharmacas, and so on. There's a huge difference between countries, and this can start at once. But to be honest, maybe it's also a measure of it's also a question, a matter of time in terms of education. And for that, we also already, together with some other institutions, we developed already teaching courses for medical doctors and pharmacists, that they have the knowledge, and that's brought into their education, that they have a different education, they know about the problem and possible approaches how to reduce it and to solve it. But that will take time. You know, education is normally some years, but also practitioners should be trained, and we did this also already very often. And yes, I think also, as you have seen from the results of Tim, there we have some opportunities to reduce at least the input at once, I would say, and then the reminder that is needed in any case, then we can go for the complete mineralization of compounds, and that will take, until in the broad, I would say, several years, maybe a decade, because that's also linked to different mind setting, that you start abandoning the idea of pharmaceutical active ingredient has to be stable for a long time. It has to be stable for a few days, maybe a few hours in your body, but not more.
And another quick question before we move to team. Ozonation has not shown any genotoxic activity in STP effluent in Switzerland in the micropole project. Do you have other results?
I do not have other results, but the question is how is the degree of dilution if you come up with one compound and you have 5 following up compounds and one of these is active, then it's good news is maybe it's only 20%, but we have no idea how these compounds behave. And I think that genotoxicity is such an important thing and property that we should reduce it or avoid it anyway. For me, it's not about acute toxicity. For me, it's about sustainability in the long run, to have clean water in the long run, and not to accept, okay, we can remove a certain share and the other is still going into the environment.
Thank you, Professor Kümmerer. I will come back to you in case
Welcome.
we have time, because there are a couple of questions more for you. But I will move now to team. The first question for team is the following. What are the PEC, quotients of ICM in the rivers?
Yeah, that's a good question. As I said, that we have some threshold concentrations, for example, for drinking water. But for the environment, it's a bit different. So the PNEX, which we have for most of these contrast media, they're quite high. So you can, some even don't have one, and there are no environmental quality standards by the European Union, because the contrast media, they were designed not to absorb to anything and to be very stable. And that's why they also, they don't cause too much harm in the environment as far as we know. But again, also what Klaus Kümmerer said, there are also transformation products, metabolites, and so metabolites not so many, but not so many that we know of, at least because we have very high excretion rate. But in the end, for the environment, it's of course the PNEX are not very high, but we still don't want those chemicals in the environment, I think.
Thank you, team. The next question is the following. You mentioned there being a health-related indicator value of 1 microgram per liter for ICMs in drinking water. Are there any limits for emissions from wastewater treatment plants or hospitals?
No, there are no, none that I know of. There are now some. There's, for example, the Competence Center on Micropollutants of North Rhine-Westphalia, here of the state in Germany, and I'm part of that. And there we are currently working on a list of concentrations, but it needs to be, specified for each wastewater treatment plant, because it depends on, how many people are connected to it, are hospitals connected to it, are radiological units connected to it. So I think it's very difficult to come up with one, with one list, which includes, just a feasible amount of different compounds, which they need to test every given time, and that makes it so difficult. But I think it should be in the, in the regulatory that at least 1 or 2 of these compounds, which are the most, for example, difficult ones, like iomeprol, for example, that they get on a list if there are lists on organic pollutants which need to be tested at the wastewater treatment plant's effluents.
And one last question also for team. So active pharmaceutical ingredients from hospitals are not even 10% of the whole API consumption. What about over-the-counter active ingredients with the urine bags? You only collect a small fraction of active pharmaceutical ingredients.
Yeah, that's a good question. Because in the end, why did we choose those ICMs? It's because you only take them for a short time, and you know that after several hours, 6, 7, 8 hours, they will be gone out of your body. So you have a very short time frame where you need to look into the excretion. Whereas for over-the-counter medication, you might take them for a longer time. You just take one pill and maybe one later, and there in these other pharmaceuticals, you often have higher metabolization of the compound. So that means that you have a lower excretion rate of the parent, at least the parent compound. That makes it difficult, but it might be relevant if you look at, for example, if you have to take a pharmaceutical which is very, very harmful to the environment, which you know of, for example, and which is causing problems. So there, and you only take it for a very short time period, there it would make sense to do that on a larger scale.
Thank you very much. We have a few more questions, but I would kindly ask our participants to follow up with our speakers. You can find in this slide their email addresses. I would like to thank the speakers for their great work and inputs today. I would also like to thank you all for choosing to join us. I look forward to continuing the conversation on pharmaceuticals in the environment in our future events. Don't forget to follow us on noharmeurope.org. I wish you a nice day and goodbye. See you soon.